For those who don't know me, I'm Lauren Moore. I'm the Chief Scientific Officer at the National Ataxia Foundation, and I'm very excited today to introduce our next speaker in our Science Showcase webinar series, Dr. Hannah Shorrock, who is a recipient of our 2020 Postdoctoral Fellowship Award. Today she'll be presenting on a CAG expansion selective small molecule screen for multiple spinocerebellar ataxias. So before we get started, just a note that you are able to turn on closed captioning by using the CC icon on your Zoom menu. And also throughout the webinar, feel free to chat to other attendees using the chat feature. However, if you would like to submit a question for our speaker to be answered at the end of the session, you must use the Q&A feature that's in your Zoom menu. Any questions will be answered at the end of the presentation. If you would prefer to give the question live during the Q&A segment, you can also raise your hand to request to be unmuted, so you can ask your question live. So a little bit about our speaker. Dr. Hannah Shorrock is a research scientist in the lab of Dr. Andy Berglund at the RNA Institute at the University of Albany. She received her Master's in Science in Biomedical Sciences and Synthetic Organic Chemistry from the University College of London. In 2018, she graduated with a PhD from the University of Edinburgh, where she studied mechanisms of disease pathogenesis in SMA. And following her PhD, she worked with Dr. Laura Rainham at the University of Florida as a postdoctoral researcher, where she received her NAF postdoctoral award. So during this time, Dr. Shorrock transitioned to studying mechanisms of disease pathogenesis of the CTG-CAG expansion disorder spinocerebellar ataxia type 8. Dr. Shorrock joined the Berglund Laboratory in 2020, where she studies shared mechanisms of disease pathogenesis and small molecule therapy development across the SCAs caused by CAG repeat expansions. Dr. Shorrock has also been a very active writer and editor for SCA Source since 2018. So without further ado, I want to introduce our speaker, Dr. Hannah Shorrock. The floor is yours.
Thank you, Lauren. Is that all good?
Looks great.
Perfect. So thank you for that lovely introduction. And as Laura said, today I'm going to talk to you about work that was originally funded by the National Ataxia Foundation Postdoctoral Fellowship on a CAG expansion selective small molecule screen for multiple spinocerebellar ataxias. And so I'm just going to provide a little bit of background. It's kind of a rationale as to why I got interested in this particular small molecule screening approach. So I'm sure as many of you know, spinocerebellar ataxias are a group of more than 40 autosomal dominant neurodegenerative diseases, and these are characterized by progressive ataxia, so a loss of balance and coordination, including slurred speech. And this predominantly arises due to damaged cerebellum, such as cerebellar atrophy, and damaged cerebellar Purkinje neurons, but a variety of other brain regions are affected to a greater or lesser extent across these ataxias. Now, spinocerebellar ataxias are genetically heterogeneous, but a large subgroup of them are caused by CAG repeat expansion mutations. So, for example, SCA12, which is caused by the CAG repeat within the 5' UTR, or SCAs 1, 2, 3, 6, 7, and more, which are all caused by CAG repeats within the coding region of the disease-associated genes. Now, CAG repeat expansions are one of the most frequently seen mutation type across the spinocerebellar ataxias, and they're also causative for some of the more common of the spinocerebellar ataxias. Now, when we look at the repeat lengths that are pathogenic for these SCAs, what we can see, with the exception of SCA6, the majority of these CAG SCAs have an overlapping range of CAG repeat lengths that are pathogenesis, suggesting that if we can target repeats in this pathogenic window, we might be able to develop therapies for multiple of these CAG expansion SCAs. And so when we think about therapeutic strategies for the CAG SCAs, clinical care really currently focuses on managing symptoms. Now, there are therapeutic strategies that target general neuroprotective pathways, which offer kind of hope across multiple different spinocerebellar ataxias, but typically preclinical therapy development has largely focused on disease-specific approaches. So whether that's a gene-specific ASO that's aiming to reduce expression of that particular CAG expansion SCA disease transcript, or whether we're looking at small molecules that try and reduce or clear polyglutamine proteins, typically these are occurring in a disease specific disease-independent manner. And what I'd just like to highlight at this point is that when we started this project back in 2020, and I believe still now, no small molecules have actually been published to reduce the expression of CAG expansion transcripts. And so what we wanted to do when we set out with this project was to take advantage of these shared CAG expansion mutations to investigate whether we could specifically target those CAG expansions using small molecules with the potential to provide efficacy across a range of spinocerebellar ataxias, including some of the rarer CAG SCAs that have less preclinical therapeutic focus. And so to do this, what I did was we designed two different constructs, one with this CAG60 repeat here, which has a nanoluciferase and then a unique sequence to allow for probe binding during qPCR. And we also have a zero repeat control here with a firefly luciferase and a unique sequence again for that qPCR. And so we integrated both of these constructs into the same HEC293 T cell genome using the piggyback transposon system. And so this allows us within a single cell or a clonal cell line to be able to look at differences between the expression at the protein level using this dual luciferase assay and at the RNA level using a multiplex qPCR to see if we've identified compounds that are regulating expression from this CAG repeat here. And so what you can see down here in B is that we generated 3 single cell sorted clonal cell lines here, so clone 10, clone 15, and clone 37, that express both our CAG60 and our CAG0 integrins. And so we can see here that clone 37 has higher expression of both of these integrins compared to clone 10 and clone 15. And we can also see this using the dual luciferase assay here, where you can see that clone 37 has kind of much higher expression of both integrins than clone 15. Again, this is kind of to further validate this protein expression, we ran Western blots for these. And you can really see via the Western blot for clone 37 that we can see this firefly luciferase here tagged by the MYC, indicated by this arrow. And by this line, we can see the MYC tagged nanoluciferase, which has this CAG60 repeat here. Now for clone 10, which is our medium expressing clone, and clone 15, a low expressing clone, you can see that we lose the ability to detect these proteins via Western blot, despite the fact that we can readily detect these via luciferase assay. We next wanted to understand more about how these cells behaved, and we noticed that clone 10, so our medium expressing clone, and our high expressing clone, clone 37, showed a significantly reduced growth rate when compared to parental HEC293 T cells. And so when we looked at toxicity, accounting for the fact that there would be fewer cells there normalizing to viability, we actually saw a 25 to 50% increase in relative cell toxicity for our CAG expression clonal cell lines compared to HEC293 T cells, which is what we would expect given the fact that we know CAG repeat expansions are heavily associated with neurodegenerative diseases. To further understand more about how usable these cells are for modeling CAG expansion diseases, we performed a transcriptomic analysis. And what we saw in line with many mouse models of CAG expansion SCAs was that we predominantly saw a downregulation of gene expression with fewer genes showing up regulation in these CAG clonal cell lines. And what you can see when we ran gene ontology on these here is that we actually saw many of these differentially expressed genes recurring in disease-relevant pathways, such as neuronal system, neuroactive ligand receptor interaction, and neuron projection morphogenesis. And we thought this was quite striking, given that the cell line is a human embryonic kidney line, and that we were seeing these changes that are kind of in disease-relevant pathways, the CAG SCAs. What I think was even more striking for us was the amount of overlap that we saw with genes that had previously been published to be differentially expressed in CAG SCA mouse models. So what you can see on this heat map here is a number of genes, all of which have been previously reported to be differentially expressed in CAG scar mouse models, which are differentially expressed in both clone 15 and clone 37 here. And we selected a handful of these that are involved in ion channels, membrane trafficking, and the cytoskeleton. And we were able to validate via qPCR over here that these genes are indeed downregulated in both clone 15 and clone 37. So now that we were kind of happy that we were recapitulating kind of phenotypic and transcriptomic hallmarks of CAG kind of repeat expansions, we next needed to identify a suitable positive control so that we could actually use this system for small molecule screening. So as I mentioned previously, no small molecules have previously been published to regulate expression of CAG expansion transcripts, but a CAG targeting antisense oligonucleotide is actually in clinical trials for multiple CAG scars and Huntington's disease. And so we used a similar sequence here to develop a CAG targeting siRNA. And so when we used this in both clone 15 and clone 37, we saw a significant reduction in expression of the CAG60 expansion transcripts and then luciferase type polyglutamine proteins compared to that zero repeat control allele when we treated with the CAG-SI compared to the control SI. And you can see that at the RNA level in clone 37 and the protein level in clone 15 and 37, the Z prime scores that we saw for these assays indicated that this is a robust assay for small molecule screening. Now, because our small molecule libraries would be in DMSO, we also did still need a positive control that we could use in DMSO for this screening system. And so to identify a positive control, we performed a limited screen of small molecules that have been previously shown to regulate the expression of other repeat expansions in different microsatellite expansion disease contexts. And from this, we identified colchicine as being able to regulate the expression of these CAG expansion transcripts, both in clone 37 and in clone 15, and again, with a Z-score indicating an assay suitable for small molecule screening. To further validate that colchicine was a suitable positive control, we went on to test colchicine in patient-derived fibroblast lines from SCA1, SCA7, and then two distinct SCA3 fibroblast lines. And so what we could see here, that in SCA1, we saw a significant reduction in ataxin-1 expression at 0.1 nanomolar of colchicine. SCA7, we were seeing this reduction at 2.5 nanomolar. And then for both SCA3 lines that we looked at, we saw a reduction in ataxin-3 expression at 5 nanomolar. And these concentrations all fall within the viability window of this compound, and we only started to see a reduction in cell viability with only a significant reduction in control 2 at this 10 nanomolar concentration. Yeah. The idea behind this screening system is to try and identify compounds that are selective for that CAG repeat expansion. And so we wanted to understand if we were also going to be affecting the expression of control alleles. And so at the doses where we saw this selective reduction in the ataxin transcripts in these scar fibroblasts, we went to look at the same ataxin transcripts in control fibroblast lines at these doses. And so what you can see here in 3 distinct control fibroblast lines, at the dose where we see this 25% reduction in ataxin-1 expression, we see no significant effect on ataxin-1 expression in these control lines. And that's a very similar story for ataxin-3 expression here. Interestingly, when we looked at ataxin-7 expression at this 2.5 nanomolar dose, what we actually saw was an increase in the expression of ataxin-7. And so this suggests that perhaps there's differences in how ataxin-7 transcript expression is regulated compared to ataxin-3 and ataxin-1. But overall, what we're seeing here is that we're seeing this reduction in expression of these ataxin transcripts only in these CAG expansion containing cell lines, and that we're not affecting those normal alleles in the control cell lines, suggesting that the screening cell line is able to identify compounds that are repeat selective. So we next went on to perform a small molecule screen of 1,584 structurally diverse small molecules. And from our initial screen, where we were looking for an effect at the transcript levels in clone 37, we had quite a high hit rate with 72 compounds that we then went on to rescreen. And we rescreened these at 2 different doses in both clone 37 and clone 15 at the RNA and at the protein level. And so here what you can see in clone 37 is that most of these compounds that we identified as being able to reduce expression of CAG expansion transcripts are able to reduce expression of polyglutamine proteins at least one of the 2 doses tested. And this compound here highlighted in red was our best performing compound in this particular assay. When we look at the protein levels of clone 15, we actually see that far fewer compounds are able to reduce the expression of polyglutamine proteins below the CAG targeting siRNA effect. But we had 3 compounds here that clearly did. And so these were HIT1, HIT2, and HIT3. And so because HIT2 was our top performing compound on protein expression reduction in clone 37, and actually outperformed both HIT1 and HIT2, but HIT1 and HIT3 by more than 20% in the original screen, we selected HIT2 to move forwards with the study. So there's very little data available for HIT2 in cell culture assays. And so before we started to look at a dose window, a dose range, we wanted to define a region at which this compound didn't affect viability in the screening cell line. And so what we can see here is that up to 1 micromolar in parental clone 15 and clone 37, we don't see an effect on viability. But by 5 micromolar, we're starting to see a significant reduction in cell viability by 20% or more. And so all of our studies in the screening cell lines, we use doses from 1 micromolar and lower. I'd also just like to highlight another kind of property of HIT2. So HIT2 is only soluble in DMSO and methanol. And so beyond 25 micromolar with 0.1% DMSO in cell culture media, we actually start to see this precipitate out. And so 25 micromolar is kind of our maximum dose that we've been able to generate data for in cell culture systems. So when we looked in this viable dose range window, what we saw in clone 15 was about a 20% reduction in CAG 60 expression relative to the CAG 0. When we went to look at the polyglutamine protein, we saw a greatly enhanced effect with a much greater reduction in the expression of these polyglutamine proteins. And that pattern was recapitulated by clone 37, where we saw this quite striking effect of 50% reduction in polyglutamine proteins here in clone 37. Again, we next went on to determine a viable dose window for treatments in our fibroblast cell lines. And so here we were able to increase the dose up to 10 micromolar, but we actually saw that these HIT2 had an effect at a much lower dose than this. So in SCA1 fibroblasts, we were seeing quite a striking significant reduction in ataxin-1 expression levels at 100 nanomolar and 1 micromolar. And in our SCA3 fibroblast lines, we were actually starting to see an effect at 1 nanomolar through to 10 and 100 nanomolar. And again, we did do these treatments in control fibroblast lines for these doses where we saw these effects with HIT2 on these transcript levels. And again, we didn't see any change in the expression of ataxin-1 or ataxin-3 transcript levels in the control in 3 different control fibroblast lines. Now, being in a lab that spends a lot of time thinking about myotonic dystrophy, which is caused by a CTG repeat expansion of the DMPK gene, and especially having identified that colchicine, which was previously identified from a DM1 screening-based platform, was able to regulate expression of CAG expansion transcripts, we wanted to understand if HIT2's effect was specific to CAG repeats or if it would also have an effect on CUG repeats. And what we actually saw when we treated DM1 myotubes was we saw a modest but significant increase in expression in DMPK. And when we looked at hallmark splicing events, in DM1 myotubes. So here we can see that from the control myotubes to the DM1 myotubes, we're seeing quite a difference. But there's absolutely no change either improving or worsening this phenotype with HIT2 treatment. And so this really suggests that the effect of HIT2 that we're seeing is specific to CAG repeats, and that we don't rescue or indeed exacerbate any CTG dependence splicing changes with HIT2. Now, because HIT2 was well tolerated in our fibroblast lines, we went on to perform a mouse model study. And so for this, we used the Ataxin-1 154Q knock-in mouse model, and we did a 2-week treatment where we were injecting 25 mg per kg of HIT2 in less than 4 ml per kg of DMSO. And we did 5 days of IP injections, followed by 2 days off, and another 5 days of IP injections. And the mice were harvested, sacrificed and dissected on the afternoon of the last IP injection. And we performed, we weighed the mice and performed recorded videos for scoring clasping 3 times a week during this treatment study, which ran from 18 weeks to 20 weeks of age in these mice. And so what you can see here is that during this 2-week treatment, none of the mice on the study actually gained weight. We saw generally a kind of plateau in terms of the weight of these mice. So we weren't seeing significant weight loss, but we would normally expect the mice to be gaining weight slowly during this time period. But what was quite nice was that we didn't see any exacerbation of this by HIT2. So this suggests that treating the mice with this amount of DMSO alone has this effect. We were also pleased to see that treatment with HIT2 didn't exacerbate the clasping phenotype in these mice. As I said, there's very little data available for HIT2 in cell culture assays, but there's no data available for HIT2 in mice. So we were really pleased to see that that our mice were actually tolerating treatment of HIT2 at this dose. So we then went on to look in the cerebellum to see if we were seeing an effect of ataxin-1, an effect of HIT2 on the expression of these ataxin-1 transcripts in this mouse model. And so what we saw here in the cerebellum of these mice, this 2-week treatment regimen, we saw about a 20% reduction in ataxin-1 expression in our HIT2 treated SCA1 mice relative to the DMSO treated SCA1 mice. And based on some sequence differences between the 2q allele and the 154q allele in this mouse model, we're actually able to design qPCR primers that specifically recognize either one or the other of these transcripts derived from these alleles. And so what you can see here when we look at the ataxin-1 2q allele specific qPCR primers is that our ataxin-1 knock-in mouse model show roughly 50% the expression of the wild type mice, which we would expect given that the wild type mice have 2 of these 2q alleles. But when we look between the HIT2 and the DMSO treated SCA1 mice, we saw no significant difference in the expression of ataxin-1 2q transcripts. When we looked at the 154q specific qPCR primers, however, what we saw was this 20% reduction in the expression of ataxin-1 154q transcripts, suggesting that HIT2 is specifically regulating expression of these transcripts through the presence of that repeat expansion, which goes back to the idea that here we're really looking for small molecules that have the potential to regulate multiple different or expression of multiple different CAG repeat containing transcripts, and that they can do this in a repeat selective manner. Now, as I mentioned, this was the first time that HIT2 has been used in mice, and so we performed H&E staining for liver, kidney, spleen, and brain, and looked at kind of general pathology, and we didn't see any kind of gross morphological changes in any of these tissues. And so we actually had the liver was assessed by a board certified pathologist. And so the only metric that they looked at that showed any changes was the liver inflammation severity score. And so here what we can see is that one mouse for each of the DMSO wild type cohort and the DMSO ataxin-1 SCA1 mouse model cohort showed slight inflammation of the liver, but we didn't see this with the HIT2 treated mice. And so this again really suggests that the treatment with HIT2 has no negative impact beyond what the treatment with the DMSO alone would have. And so this was really promising. And so it suggests, that HIT2 is overall well tolerated and doesn't induce any significant morphological changes in vivo. Being from working in the RNA Institute and spending a lot of time thinking about transcriptomics, we also wanted to understand if HIT2, which we know is regulating the expression of these ataxian transcripts at the RNA level, whether it's having any other effects at the level of the RNA. So whether that might be off-target effects or whether we could actually be looking at any potential rescue of transcriptomic hallmarks is the next step that we wanted to look at. And so before I kind of get into that data, I'm going to transition briefly away from our HIT2 small molecule story, talk to you about some work that we've been doing looking at transcriptomic changes in CAG expansion spinocerebellar ataxias. And this work really comes from kind of my motivation and my interest in understanding overlapping mechanisms of disease pathogenesis and how we can leverage this not only for cross-disease therapy development, which I've been talking to you about so far, but also for potential cross-disease biomarker development. And so looking through the literature, we see a lot of transcriptomic changes, but it seems like there's really kind of a lack of consensus of these pre-symptomatic and potentially disease-driving transcriptomic changes. And so I was interested to understand if we could identify any transcriptomic changes that we think might be really relevant to disease pathogenesis. And so I set out to form comprehensive analysis of RNA sequencing data sets from CAG expansion SCA mouse models, which ended up being from SCAs 1, 3, and 7. And while I did originally start out looking at differential gene expression, working in a lab that studies myotonic dystrophy and spends a lot of time looking at alternative splicing, this actually rapidly changed into a study looking at dysregulation of alternative splicing. And so just kind of very briefly, when we think about splicing, we have constitutive splicing, which is where the introns are removed from the pre-mRNA, and we join the exons together to form the mature mRNA. Alternative splicing is where we're processing these RNA transcripts into distinct combinations. And this can be regulated in a developmental or temporal manner or a spatial manner, leading to production of potentially different protein products in different brain regions, for example, or at different stages of development. And we know that alternative splicing is dysregulated across multiple microsatellite diseases, including the CAG expansion disease, Huntington's disease, and perhaps most notably in myotonic dystrophy, where alternative splicing has been really well characterized. We know that misspliced events have been linked directly to key phenotypes of this disease. And so we set out to form a comprehensive analysis from publicly available RNA sequencing data sets. And so we did a comprehensive search using GEO and NCBI, and we identified a range of data sets that met several criteria and were included in this analysis. So today I'm briefly going to talk to you about the data from the cerebellum, but in the paper that we published on this last year, we also look at data sets from a variety of other brain regions of CAG expansion SCA mouse models. So here we're looking at data from the 82Q transgenic SCA1 mouse model, the 154Q SCA1 knock-in mouse model, two different mouse models of SCA3 and a model of SCA7. And when we look across all of these data sets, we do detect dysregulation of alternative splicing, with dysregulation of these cassette exons or skipped exons in dark blue being the most predominant type of splicing dysregulation that we were seeing. And when we look at the changes that we're seeing in each of these data sets, so this is for skipped exon events, and it's the size of the change of these splicing changes that we're seeing, you can see that we're not just seeing small changes. Some of these changes are quite sizable, suggesting that this could be having an impact on the phenotype. When we look at the genes that these splicing changes are occurring in, so here I'm showing you gene ontology enrichment analysis from genes that show skipped exon misregulated across two or more data sets. What we can see is that we're seeing enrichment of disease-relevant pathways for the CAG SCAs. So things like regulation of synapse organization, actin filament-based processes, neuron projection development, regulation of membrane potential. We know these are all kind of relevant pathways when we think about the spinocerebellar ataxias. We can also look at this in a different way. So here we've performed gene ontology enrichment analysis on each data set individually, and we're showing a conglomerate depiction of this, where each data set is represented by a distinct color, and each individual circle here represents a different gene ontology term. And so we can see again that kind of specific disease relevant terms coming out here. So we've got a cluster of terms here that are related to ion channels, that are related to the synapse. We have a range of terms that are related to cytoskeleton and neuron projections here, as well as nuclear processes, which includes things like transcriptional regulation and DNA damage and repair. We also wanted to understand whether these changes were, or to define whether these changes were kind of dependent on that repeat expansion, or whether they might have been caused by how those mouse models were generated. And so for 3 of these data sets, we were actually able to look at a comparison between the pathogenic repeat length mouse model versus a mouse model that was generated in the same way, but using a repeat length that was in the normal range. And so when we did that comparison, and here we're looking at 1 SCA1 data set and 2 SCA3 data sets, and we still saw a similar number of dysregulated skipped exon events. And again, the magnitude of those changes that we were seeing here was similar to what we were seeing when we were comparing them to wild-type mice, suggesting that this is dependent on those pathogenic length repeats. We were also interested to understand the relationship between differential gene expression and alternative splicing. And so here I'm just showing you 3 data sets where we have a pre-symptomatic SCA1 and a pre or early symptomatic SCA3 data set, then quite a late symptomatic SCA3 data set. And what we can see in these 2 data sets from early in disease pathogenesis is that we see far more of these skipped exon events than we do differentially expressed genes. But much later on in disease, we see far more of these differentially expressed genes. And while I'm not showing you the data today, this was a consistent effect that we saw across pre-symptomatic data sets, suggesting that not only these splicing changes repeat length dependent, but they are also a pre-symptomatic transcriptomic hallmark of these CAG SCAs. And so we then went on to identify individual splicing events that were implicated in some of these disease-relevant pathways, and that we saw some of these repeat length and pre-symptomatic dependent changes. And so here I'm showing you an example for TRPC3 exon 9. So when we look in cortex and brainstem regions, exon 9 is almost completely included in these regions. But when we look in the cerebellum in the wild-type mice here in dark blue, or this pale orange short repeat length mice, we see that this exon is only included maybe 25 to 40% of the time. And what we're seeing in the SCA mice is we're seeing a shift towards the level of inclusion of exon 9 that we would see in other brain regions. And so, in other words, what we're seeing is basically a reversal of this cerebellar-specific splicing profile for this ion channel. And we were able to validate this in 12-week and 23-week Ataxia 1154Q, 2Q, SCA1 mice using RT-PCR. And the thing that was really kind of quite exciting about this particular event that I've chosen to show you today is that the difference in the splicing has been well studied in terms of the function of the TRPC3 ion channel. And so we know that increasing exon 9 inclusion actually leads to a reduction in membrane conductance. And so this is something that we're seeing pre-symptomatically in these data sets that could be having actually an effect on how these neurons are functioning. And so in this kind of brief interlude from the small molecule story, what I've told you is that alternative splicing dysregulation occurs pre-symptomatically in disease-relevant pathways and in a repeat length dependent manner in SCA1, 3, and 7 mouse models. And I've also shown you that we have identified alternative splicing of ion channels with known functional consequences in genes with pathogenic links to SCA. So the example that I showed you, TRPC3, we know that mutations in TRPC3 cause SCA41, but we also identified a range of other skipped exon events in a variety of different genes that are known to cause different subtypes of spinocerebellar ataxia. And while I didn't show you the KCNMA1 example today, this is another case where the function of that particular splicing change that we see in these data sets is known to have a functional consequence on the ion channel itself. And so when we think about transcriptomic changes in the CAG expansion SCAs, and we're looking at whether HIT2 is capable of rescuing those, we're particularly interested in understanding if HIT2 can rescue alternative splicing dysregulation in the SCA1 mouse model that we looked at, especially given that it is regulating the CAG expansion transcripts. Now, as I mentioned before, all those data sets that we looked at were publicly available RNA sequencing data sets, and they were generated using a poly-A selection-based library preparation method. Now, typically in the lab for myotonic dystrophy-based work, our standard protocol is to use an RNA depletion-based method for library preparation. And so here, this is in-house generated RNA sequencing from our cohort of mice that were treated with either DMSO or HIT2. And so what you can see is that we're able to independently validate splicing changes where we are seeing a predominance of dysregulation of skipped exon events in the ataxin 1154Q mice, and that the changes we're seeing, we are seeing some substantial changes. We're not just seeing small changes. And again, these changes are in disease-relevant pathways like nuclear localization, regulation of synapse organization, calcium ion-regulated exocytosis of neurotransmitter, presynaptic depolarization and calcium channel opening, as well as more cytoskeletal associated terms such as actin filament-based processes as well. So now that we've in-house validated these splicing changes using kind of an independent library preparation method, we next went on to try and understand the effects of HIT2 on the transcriptome. And so while I am going to focus on looking at skipped exon events, we did also look at differentially expressed genes. And so just to orient you to this plot, what we're looking at is our DMSO-treated SCA1 mice versus the wild-type mice on the x-axis, and on the y-axis, we have our HIT2-treated SCA1 mice versus our wild-type mice. And so here, what we can see in blue is that we actually see relatively few differentially expressed genes between our wild-type and our SCA1 mice, with most of these clustering around this dotted line, which indicates that, generally these genes aren't rescued, with some of them showing mild rescue here. And this is really reflected in this PCA plot for differentially expressed genes, where we can see some of these mice are kind of trending towards where the wild-type mice are, but generally there's no distinct clustering between the DMSO and the HIT2-treated SCA1 mice. What is very notable from this plot, though, is that we only actually saw 4 genes that demonstrated an off-target effect where they were differentially expressed between our ataxin 1 HIT2-treated mice and our wild-type mice. And so this suggests that HIT2 is having a very low impact on, a very low off-target impact on differential gene expression. When we look at the same plot for skipped exon events, again, we can see here that we have the phenotype events in this blue and our off-target events in pink. And while we do see more of these off-target events for the skipped exon events, this only actually represents 0.59% of all the skipped exon events that were detected in this data set, again suggesting that HIT2 is actually having a reasonably low off-target effect on the transcriptome. When we look at the phenotypic skipped exon events, where we have a significant difference between our ataxin 1 mice and our wild-type mice, we can see that we're seeing quite a lot of events here that are dysregulated in these SCA1 mice, and with many of these actually clustering along the x-axis, which indicates that these are kind of largely rescued. And so this is really quite clear from the difference between the PCA plot, where when I showed you the differential gene expression plot, we weren't seeing much rescue. With the plot for the skipped exon events here, we can see that our HIT2 treated mice are sitting roughly 50% of the way between our wild type mice and our DMSO SCA1 mice. And so this is suggesting that we're getting really quite nice rescue of these skipped exon events. And when we look at this, so we have 495 events in this data set that are dysregulated between wild type and ataxin 1 DMSO treated mice. Of those, more than 350 events are rescued with kind of a minimal number of events showing no rescue or an opposite effect. And when we drill down into these events, what we can see for the events that are rescued, so if you look here, from the DMSO treated SCA1 mice to our HIT2 treated SCA1 mice, we're seeing a shift of the inclusion of these events almost back to kind of an average around where the wild type levels would sit at 0% for the inclusion events. The exclusion events, we don't seem to see quite as good a rescue overall, but we're still seeing a shift towards where we would see this for wild type mice looking on average. Now, driving down into this a little bit more again, here we're looking at individual events that are significantly rescued in comparison to the DMSO treated mice when we have HIT2. And so what you can really see from this plot is that we're seeing these HIT2 treated mice here showing a much more similar overall splicing profile to the wild type mice here than they do to the SCA1 mice treated with DMSO. And so we can see that with a few select events here that I'm showing you. And we've been able to validate these this rescue of splicing for this event here, CACM2D2 exon 23. And so just to provide you with kind of a summary of what I've shown you today. So with the postdoctoral fellowship from the National Ataxia Foundation, really set out to develop a CAG expansion selective small molecule screen for multiple spinocerebellar ataxias. And so what I'm hoping I've shown you today is that we now have proof of concept that we can use small molecules to selectively reduce levels of CAG expansion transcripts across model systems for multiple different CAG expansion spinocerebellar ataxias. To do this, we developed and characterized a CAG repeat selective screening system that we showed replicated phenotypic and transcriptomic hallmarks of CAG expansion SCAs. From a screen of 1,584 compounds, we've identified 3 structurally diverse small molecules that do regulate CAG expansion transcript levels. Showed you that HIT2 reduces levels of CAG expansion transcripts in SCA1 and SCA3 patient-derived fibroblasts, and that it also selectively reduces levels of expanded ataxin 1 transcripts in an SCA1 mouse model, where it also rescues the dysregulation of alternative splicing. I've also shown you that HIT2 is well tolerated, induces minimal off-target transcriptomic effects and minimal morphological or inflammatory changes in terms of kind of systemic toxicity evaluation. And so with that, I'd like to thank my mentor, Andy Berglund, as well as the many people who worked on both the small molecule story and the splicing story that I kind of talked to you about as well. I'd also like to thank John and Carlock at the RNA Institute, who really helped with the kind of original design of the system that we've developed, and then Damien Shin at Albany Medical College, who we work with to do the mouse work on the ataxin 1 154Q knock-in model. And I'd like to also thank the funders. So based off of this National Ataxia Foundation postdoc fellowship, we've also now been able to kind of leverage these data to secure federal funding for our alternative splicing and SCA small molecule research projects as well. And with that, I'd just like to thank you all for listening today, and I'm happy to take any questions.
Well, thank you so much, Dr. Shorrock, for that wonderful presentation and for all the hard work you put into this ataxia research over the last couple of years. So I have a question for you. I know you're not naming the HIT2 molecule, but can you tell us a little bit about whether you think that molecule is actually a good candidate for development into potential treatments, or if it needs to go back into some reformulation?
Yeah, so I think that's a great question. So HIT2 is not soluble in anything other than DMSO or methanol. And so currently at the moment, we are working on structure activity relationship studies to both try and improve the solubility and kind of improve the effects that we're seeing on the CAG transcripts and minimize any potential effects that we were seeing on those CAG transcripts that I showed from the myotonic dystrophy myotube model.
And did you test them on any non-CAG but ataxia specific disorders?
No, we haven't actually. I do have some fibroblast lines from non-SCA ataxias that I could potentially look at. So yeah, I think that's the mechanism of action, or what we think the mechanism of action of HIT2 is doing. We think it's kind of specifically affecting the CAG repeat. And so I wouldn't expect to see a change in the kind of disease-relevant genes in those cell lines, but it would be a good place to gather more information about off-target effects.
Mm-hmm. Yeah, we had one question specifically from the audience about FA, which I think is a good example of a repeat expansion that's not a CAG, but is ataxia specific. So
yeah, I think that's quite interesting. I think, here we're looking at trying reducing expression, and so it'd be curious to see if we kind of what the effects would be in an FA model, for example.
Another question about the specific molecule you're working on. How do the small molecules or their analogs, what is their ability to cross the blood-brain barrier?
Yep, so we know HIT2 gets into the brain. We know it passes the blood-brain barrier. So the delivery route that we used here was IP injections, and following that 2-week treatment, we have been able to detect HIT2 in the brain. We haven't been able to quantify it yet, but we're still kind of trying to work on that and troubleshooting it. But it does cross the blood-brain barrier. As we move through SAR studies, that is something that we'd obviously be kind of very keen to make sure we maintain and perhaps actually improve the amount of the compound that is getting into the brain.
One question, do you know if your compounds target transcription from DNA to pre-mRNA or splicing from pre-mRNA to RNA?
So I personally think what we're seeing is binding to the CAG expansion RNA. So I don't think we're actually particularly affecting transcription here per se. One of the reasons that I think that is because of, we see some effect at the RNA level, but that does actually seem to cap out, whereas in the screening system, we continue to see a reduction in the expression of the polyglutamine proteins. Sorry, could you repeat the question again? I seem to have lost myself.
Yes, it was whether you're targeting transcriptional changes in the DNA to pre-mRNA or the pre-mRNA to mRNA.
Yeah, and so then kind of in terms of kind of the effect we see on splicing changes, so I wouldn't necessarily say we're kind of regulating the splicing of those ataxin transcripts themselves, but more that by kind of targeting those repeats, potentially binding to them and reducing their expression, would then having kind of a knock-on effect on the transcriptomic changes we see in these diseases.
I know that you saw a specific targeting of the allele containing the repeat expansion in the ataxin-1 mouse model. Were you able to see whether the ability to target and change expression changes with repeat length? So in cell lines with variable repeat lengths or mouse lines with variable repeat lengths?
That's something that I'd really love to do. It's not something that I have done yet in a controlled manner. We do know, so the control fibroblasts that we've looked at, we have sized the repeats for ataxin 1, 3, and 7 in those control lines, and the allele sizes for those are roughly similar to the non-expanded alleles that we see in the SCA patient-derived fibroblast lines. And so, we're kind of hoping that the window that we see these effects is really kind of beyond that 30 repeat range. I think it would be interesting to look at some of these compounds in, say, SCA6 fibroblasts, for example. You know, would we see an effect on expanded alleles there, or would we not, given that the repeat length is so short? And the other thing that I wonder about is whether the expression level of the transcripts, rather than just the repeat length of the transcripts, also plays a role. So, you know, if you have a lot of a particular transcript that has maybe more of a moderate repeat, would we regulate the expression of that?
If you can tell us a little bit more about HIT2. What is the approximate affinity range of these small molecules, and what's the approximate molecular weight of HIT2?
So I don't know the approximate. I don't know, maybe about 300 off the top of my head. So we're currently working on a variety of kind of assays to try and understand more about specific interaction of HIT2 with the repeat RNA. You know, the screen that we designed is really a unbiased screening approach. So, you know, we could identify compounds like colchicine that doesn't bind to RNA but regulates expression through different mechanisms. We could identify compounds that regulate transcription, or compounds that lead to degradation of the RNA, or have kind of, you know, maybe bind directly with the RNA. And so we weren't selecting for any particular mechanism of regulation specifically here. But hopefully kind of when we publish the story, there'll be a little bit more information on some of that aspects for you.
And another question about HIT2. If it isn't soluble in most substances, would a prodrug be achievable to increase the solubility for this molecule?
Yeah. I think that's definitely something that we'll keep in mind. Currently, at the moment, we're kind of trying to improve the structure of the compound itself. And another thing that, you know, we might think about is kind of alternate delivery routes. But I think a prodrug could potentially be the way to go.
Sorry, I was curious. You showed this worked this had effects in multiple repeat expansion SCAs. Was there a particular reason that you chose to move forward with SCA1 for your mouse study?
Yeah, so I suppose kind of two reasons. One, we saw the kind of biggest effect, the biggest reduction on ataxin transcript levels in the SCA1 fibroblasts that we looked at. But also kind of roughly around that time, we found out that Damien Shin had recently got an ataxin-1 154Q knock-in mouse model colony, which is 20 minutes down the road. So it was kind of a mix of happy coincidence and the data as well. So we are open to testing in other mouse models, but I think we'll probably wait until we have a slightly improved compound that we can do kind of more long-term studies with.
And I think we both know that there have been many different drug screens and cell screens performed across the ataxias. Can you just talk broadly about what extra strengths or power you think that this new screen brings to the field?
Yeah, so I spent a reasonable amount of time looking at previous screening systems, and I think, you know, there's a lot of powerful screens out there that, you know, have identified kind of FDA approved compounds and things that you know, could be really quite relevant. I think two of the things that are kind of quite a strength of the system that we developed here, and this is both really based off the back of a similar system that the Berglund lab published back in 2019 for myotonic dystrophy, but where we have the two constructs integrated into the genome of our cells, right? So each cell expresses both our zero repeat and our CAG60 repeat alleles. And so that gives us the ability to be able to do that ratiometric analysis. So, you know, when we're not selecting for things that regulate kind of different aspects of that transcript there, we're kind of really looking for things that target the CAG. And then I think And this really just goes back to kind of my research interests, where I am really interested in understanding kind of overlapping mechanisms of disease pathogenesis, and kind of how we can leverage that for shared therapy is that most of the screening systems are in the context of a particular gene. So, you know, the repeat expansion is in the context of ataxin 3 or ataxin 1 or CACNA1A. And so that gives you a lot of power for one particular disease, but it does also select for things that target other regions of that gene or that protein, other than the CAG repeat. And I think, you know, that can lead to compounds that regulate those proteins in general, so that they lose that kind of repeat selective effect. But it also means that, you know, and I don't think we can actually claim that they don't work in other diseases, because I don't know if any of compounds identified in a disease-specific screening system have been tested in models of other diseases. But it potentially also limits that transferability. And I think, you know, for me, with the spinocerebellar ataxias being so rare, if we can identify compounds that can work across multiple of those, then, you know, I think that could be quite a benefit.
And I think this last question is a great one to end on, which is what is next for this project and what is next for you?
So next for this project is kind of, you know, we have, we're currently kind of working on developing an improved HIT2. We have 2 other compounds that we are kind of continuing to follow up that are kind of structurally diverse. And so, you know, that was one of the motivations behind choosing this particular library, in that, you know, structural diversity could give us a handle for multiple different ways that we can regulate sex expression. I think, you know, I'm very interested in screening FDA-approved compounds using this platform, but we are currently kind of redesigning to develop an inducible system that will give us much more control. And then for me, I'm going to be starting my own lab at the University of Albany in the Department of Biology this fall. And so hopefully I'll be carrying on some of this research, well, across the corridor from where I am at the moment. But yeah, thank you.
Congratulations on your work, and thank you so much for your presentation today.
Thank you very much.
Thanks, everyone.